of incubation in the sodium hydroxide SDS solution, we add potassium acetate and incubate another 5 minutes. Now what does potassium acetate do? It lowers the pH. So that means it's going to allow DNA to reanneal. Okay? However, what happens before the genomic DNA has time to reanneal is it's being broke down by the proteins that are participating. Okay? However, the plasmid DNA, when it melts, it stays as a ring inside of a ring. Okay? It stays like a figure eight. So you hit it with the potassium acetate, lower the pH, and it's just going to fall right back over and reanneal. So what's going on is you're reannealing the plasmid DNA, and the genomic DNA does not have the opportunity to reanneal because after that 5-minute incubation with potassium acetate, the whole tube is centrifuged, and so what you're going to be doing is pulling this gunk of genomic DNA with the precipitating proteins and pieces of cell wall and cell membrane. And that's going to pull that genomic DNA down in pellets. The plasmid DNA is so small in size that it is not affected by the precipitating proteins or the pieces of cell debris. And so what will happen is your plasmid DNA will stay in solution until after you take the tube out of the centrifuge, you just simply collect your supernatant and you throw away the pellets. And you've got pure, literally, well, there'll be some proteins in there. You've got relatively pure plasmid DNA completely separated from the genomic DNA component. So, after we do that, all we want to do now is collect it, so we precipitate it with isopropanol and go through a couple of ethanol washes, again using 70 or 80% ethanol sometimes, not always, to dissolve out any salts that might have co-precipitated. We've got reasonably pure plasmid DNA. After we do the last ethanol wash, we will put that in the feedback and evaporate off the last bit of ethanol. That takes, depending upon how many tubes are in there evaporating off the last bit of ethanol, it can take anywhere from 5 to maybe 10 minutes. And you're ready then to decide, based upon the size of your pellet, the size or the volume of PE buffer to re-suspend the DNA in. And usually at that point I look at the pellets and tell individuals how much PE buffer to use. Now, when you do a mini prep like this, your pellet is going to look white. And it'll look off-white until it's dry, and then it will look white. That white is not DNA. That white is protein that is contaminated, that has contaminated the prep, but usually it's not enough to affect what we're going to be demonstrating, so we don't worry about it. Pure DNA, and you will get pure DNA out of this prep. So you will have pretty pure genomic DNA when you run this prep in 2 weeks. And what will happen, I'll just warn you now, what will happen is you'll do a spin and you'll pellet it and you'll pour off the ethanol and you'll see end of the test. And you will say, I did not spin anything. And I will, if you're in my lab, I will take the tube and I will hold it up to the light and I will look at it and I will say, yes you did. You just can't see it. Because pure DNA does not have a color. It is not white. It looks like dried, it is dry. Uncooked egg white. Okay? It's translucent. Or another way to put it is just like clear straw. But that's what it looks like. And so it's hard to see when you're isolating small amounts of it, but you'll get it out. It's all so small. Okay, any questions about that? Okay. Then let's go on to the eukaryotic. Now, in eukaryotic systems, as I have said already, if you are isolating the DNA program, you have multiple compartments in the eukaryotic cell. Unless you're working with a single-celled eukaryote, you're really not even working with single cells. You're working with cells that are aggregated with other cells that we call tissue. Okay? So what we do is, again, you have to know what your goal, obviously, is to the isolation. Sometimes you can get to the total DNA isolation out of the eukaryotic cell. If it is an animal type of eukaryotic cell, you're going to get two genomes out. You're going to get out the nuclear genome and the mitochondrial genome. If you're working with a photosynthetic organism and you isolate DNA, you're going to get three genomes out: the nuclear genome, the mitochondrial genome, and the chloroplast genome. Now, if you are not working with the mitochondrial DNA or the chloroplast DNA, most of the time, if your questions are about the genomic DNA from the nucleus, most of the time it doesn't make a lot very different in your protocol, but you've also co-isolated organelle DNA with it. The reverse is not true, however. If you're working with organelle DNA, you've got to get rid of the other organelles, like if you're working with plant systems and you're working with chloroplast genomes, chloroplast DNA, you've got to get rid of mitochondrial DNA and nuclear DNA. And there are ways to do that, but we do it through centrifugation techniques. Okay? So, let's start with the genomic DNA again, just talk about generalities of how we're going to isolate this. First of all, since you're working with tissue and based upon a question that we had earlier today, what we do is we collect the tissue, or if they're single cells, and we flash freeze them in liquid nitrogen, and then store the tissue or the single cells in a -80 freezer until you are going to actually do the isolation. Otherwise, these eukaryotic cells get very unhappy, okay? And if they are unhappy, they will open up their lysosomes and they will release all those degrading enzymes that are stored in the lysosomes of the cell, and they will mix everything and you will get nothing usable out of it. So you have to be, you have to be careful with that. So we flash freeze. And then when we're ready to do the isolation, we get the material out of the -80 freezer and we grind it somehow. Sometimes we just have big mortars with big pestles and we just pour liquid nitrogen in a buffer system and we just grind and grind and grind. For some systems, you can even use a drill with a little stone on the end of it, literally. And for, there are various ways. Basically, it involves mechanically shearing the tissue and some of the cells. Because if you've got a tissue system, you're not going to mechanically shear individual cells by simply grinding. Okay? But you are going to disrupt the sheets of cells that are not tissue. Then what we do is we go to a strategy of centrifugation that's called differential centrifugation. And I want to just briefly give you a general idea of that. Obviously, with centrifugation. Differential centrifugation takes the homogeneous. You've added buffer, and you've ground some more. That homogeneous, and you put it into a tube. And if you think in terms of the pieces parts of the cell, the organelles, the mitochondria, the chloroplast, the membrane systems of the endoplasmic reticulum, individual chemicals that might be out in the cytosol, like proteins, tRNA, individual other structures that are not membrane bound, like ribosomes, for example. All of that is in the homogeneous. And all of that has different sizes. And so the idea of differential centrifugation is use the size, the weight of a component of a cell to pull the heaviest stuff down first and then lighter stuff down next. And the way you do that is to set your centrifugation speed. If you centrifuge at a low speed, you are only going to pull down the biggest thing. Okay? And so, if you don't want those biggest things, you do a low speed centrifugation, you pellet the biggest things. In this case, it would probably mainly be nuclei and cell membranes. And then you save your supername, put it into a fresh tube, and centrifuge it again, but at a higher speed. So the second time you centrifuge, you pull down the next heaviest stuff. And so you can go through a whole series of these kinds of spins that grossly, not finely, but grossly separate the components of the cell relative to their size. Now, when I say it does it gross, I mean, this picture looks beautiful. It's like it's separating it completely. When you do this first spin and you have nuclei down in the pellet, we don't do a long spin. So you're still going to have some nuclei up in here as well. That's why it's gross. Okay, you get rid of a lot of it, but not all of it. Okay, so that's one strategy we will often use with the eukaryotes. Then what we do when we have the portion in which we have maximized what we're looking for. Maybe we're looking for mitochondrial DNA or whatever, and we have the fraction that's going to have the most mitochondria in it. Then what we start doing is we start removing things that we don't want. So we hit it with RNases to digest the RNA molecule, then we hit it with proteinases to digest the protein. If you do that, you then have to remove the RNases and the proteinases, okay? And so we will do a phenolchloroform procedure, just like I described for the prokaryotes. Do a couple of washes of that. Once we finish our phenolchloroform washes, then depending upon the purity of the DNA that you need and the type of DNA that you are trying to collect, we will do one of two things. We will precipitate it with isopropanol, pellet it, wash it, and collect it, and go on to our research protocol. Or we will have to go to isopycnic banding type of approach. And I need to talk to you about two other kinds of centrifugation because they play into this depending upon what you're trying to understand. Okay, so the other two types of centrifugation I want to define for you is the rate zonal type of centrifugation and the isopycnic banding type of centrifugation. We do not do much rate zonal work if you're working with DNAs or RNAs or proteins. If you're working with any of those three components of the cell and you're going to have to get purified chemicals out, you're going to have to go to isopycnic banding. But you need to understand the difference between isopycnic banding and rate zonal. Okay. Rate zonal uses the same centrifuge as the differential centrifugation. It's called a high-speed centrifuge. Okay. So as you go down in the centrifuge room, that would be your Sorbol type centrifuge. The Sorbol that's in there is a high-speed centrifuge. And what rate zonal does is it uses what is called a step gradient to sort things out by size. Okay? And the step gradient, the material of the gradient, is oftentimes a sucrose gradient. And it is a step gradient because the concentration of the sucrose changes in step increments within the tube. Okay? So what you do basically is you take sucrose and you mix it up at a high concentration, and you put that high concentration sucrose in your centrifugation tube. And that is your first step of your gradient. It is your highest concentration. And then you mix up another solution of sucrose at a lower concentration, and you dribble that down the side of the tube and produce a second layer of sucrose, but at a lower concentration than this layer. This will literally float on top of that. There is that much difference in their sugar concentration. And then you mix up a third, most dilute concentration, and you dribble that down, and it will ride or sit on top of this layer. You add your liquid that has all the stuff that's left from your isolation, and you put it in a high-speed centrifuge, and you centrifuge, and the material will move through the gradient until it reaches a point where it's at equilibrium with the concentration of the particular medium. Okay? And so what you will get is bands within each or one or more of the steps of the gradient. And then you simply put a pipette down and suck off the band that you want. This kind of thing, predominantly, the step gradient approach is predominantly used to separate out different cells based on the size. And so it's very handy and convenient if you're trying to isolate red cells from white blood cells from platelets. You're doing blood work. You're going to do red cells. But it is not very helpful for us if we're working at the molecular level with DNA or RNA. For that, we go to our particular banding. If we have to purify away, we have to purify the different DNAs or RNAs away from each other. And so let's just think in terms of DNA, it's easier to think in terms of that. If we are doing an isolation from a eukaryotic tissue that has chloroplast, mitochondria, and nuclear DNA, and we have to have pure, let's say, chloroplast DNA, then you're going to have to go to an isotopic banding approach to purify the chloroplast DNA away from the mitochondrial and the genomic DNA. Now, isotopic banding, they show it here with a steep sucrose gradient. We don't do that. If we're doing DNA work, we use a different medium, and it's cesium chloride. And so we use the cesium chloride density gradient. And that particular gradient we use for ultracentrifugation is what is called a continuous gradient. And what that means is you mix up the cesium chloride at a given concentration based upon your ultimate goal for your isolation. And you don't have to just figure this out. There are tables that tell you what concentration to mix the cesium chloride up in if you want to isolate chloride, for example. So you isolate or you mix up your cesium chloride gradient and you put it in an ultracentrifuge tube. These tubes have a little knob at the top, and you use a heat seal to seal it. And there is cesium chloride all the way. I mean, you fill this up completely. There's no air left. And of course, you mix your prep that has your different DNA. You mix that in with the cesium chloride. All of that goes in here, and you seal the top. Okay? And then it goes in an ultracentrifuge, not a high-speed centrifuge, an ultracentrifuge. An ultracentrifuge pulls a vacuum while it's spinning, while the rotor's spinning. And it can give you G-forces of 400, 500,000 times the full gravity. Okay? And generally, these are long spins. 36, 48-hour spins. But what happens during the spin is that the cesium chloride self-forms a continuous gradient. And what that means is, as you move down the tube, the density of the medium increases. So it is self-forming, and it is a density gradient. And then what's going to happen, again, talking about different kinds of DNA, what's going to happen is these DNAs in the centrifugal force will move through the step gradient until they reach a point where they are at buoyant density with the cesium chloride at some position in that tube. And so if we're isolating genomic DNA from mitochondrial DNA from chloroplast DNA, what you are going to see is 3 bands develop after your 36 or your 48-hour spin. You will have 3 bands. If your concentrations are high enough, of course, you will have 3 bands like that. This is your nuclear DNA band. The second band up is your chloroplast DNA. And your top band is your smaller, least dense DNA, which will be mitochondria. So that you can separate those 3 DNAs out from each other, but the way to do that, the only way to do that, is with isotopy branding. We obviously don't do this in class, because if you have a 36 or 48-hour centrifugation spin, it's not conducive to 3-hour lab time. Anyway, after that spin is over, okay, so how do you recover your DNA? What we do is we take a 16-gauge needle from a syringe, hypodermic needle, and we stick that needle right in here so we can let air in. And then we take somewhere between a 16 or an 18-gauge beveled needle and put it on the end of the syringe. Okay? And when I say beveled needle, I mean the end of the needle is at a slant, beveled, as opposed to just cut straight off. Okay? And what you do, let's say you want to collect chloroplast DNA, and you don't want the nuclear DNA, and you don't want the mitochondrial DNA. What you do, and I should also add that we put dye in here, and we go into a dark room, and we take UV light and we shine it on here so we can see these bands, otherwise you don't even see them if you get off, okay? What you do is you stick that beveled needle. Not good at drawing this. Okay. Take the beveled needle on the syringe. And you put it in below the band that you want to collect. And then you just slowly suck that band into your syringe. Okay? And then we have to clean up the DNA afterwards, and I'll tell you about that in a few minutes, but let me just show you. This is what these tubes look like, and this is the result of a prep that a graduate student of mine and I were doing probably about, 12, 15 years ago. Anyway, we were collecting chloroplast DNA from cytosine, and it was really hard to get out there. We had very low concentration of chloroplast DNA in the chloroplast. The chloroplast were tiny little things. And so what we would do would be a total DNA isolation, and we go through the phenol chlorophyll and all that. And then we get to the cesium chloride gradient step, and we run a cesium chloride gradient. And if you come up here and get close, you can see a band there, and there's a little band here as well. What was going on is we were getting out very low concentrations of things, and so what you do with that is you simply do lots of tubes. Okay, and so we did lots of tubes. And then we would collect our chloroplast band from all the tubes that we were running during that particular day. And we would combine that material that we had isolated from this section And then we did a second cesium chloride centrifugation, and you can obviously see the band here. And basically what we're doing is concentrating the chloroplast DNA. And we pulled that band out. Now once you pull that out, you've got cesium chloride all around your DNA. So what you have to do is you have to get rid of that pulp, you have to get rid of the cesium chloride, and you just dialyze it. You put your DNA in a tube, in a tube, in a tube that has pores of a certain size in the tube wall, and you put it in distilled water, and the salt will diffuse out, and that's called dialyzing the DNA. Then you put it in a regular tube, and you state with alcohol, and you've got the DNA. So that's the approach we use with those kinds of things, okay? Any questions about that? Okay, then the one final thing I want to talk to you about that will finish up the section, and I won't get it finished today, so we'll finish at the last 10 minutes of the class on Tuesday. But what I want you to think about is what characteristics of DNA can we use if we have DNA that we don't know what genes it's got, okay, and we're looking for one specific gene, and we, besides having the DNA that we're working with, we can get a small piece of DNA that contains a portion of that gene sequence, okay? And we can use that known DNA sequence to ask the question, is this gene in my DNA? In the DNA I'm working with? What attribute of DNA can we use to answer that question? Attribute of structure of DNA. No, we're not, well, you know, in the prep you might cut it up into pieces to try and find the gene. But think at the DNA level. We have a piece of DNA that has the known sequence. And we have DNA that we don't know whether it has the known sequence. What does DNA do?
We can label RNA.
No, we're not working with RNA. We're working with DNA. We could label the DNA. So my question would be, what DNA are we going to label? Well, you don't want to label what you don't know, right? Because you don't know who's got it. Okay, so let's go with labeling what we know. Labeling the little piece of DNA that has the sequence that we know. When we do that, that little piece of DNA that has the sequence that we know is called the probe. Okay? And then we can take our unknown DNA and we can melt it, and we can have it in some kind of a solution of some kind, along with the labeled probe. Now, what's going to happen if the labeled probe finds a section of DNA that's complementary to it? Huh? Yeah, it's going to hybridize. This is the DNA hybridization protocol that I'm trying to pull out of you. It's also called probing. Okay? This is the way you go find genes. Okay, and we will start with that next Tuesday. Remember, we do not have class this Thursday. The only break is all semester.